Hello, everybody. Let's start with the science. So it's a pleasure to introduce you today to Stephanie Dutkiewicz, a senior researcher at AMT. Her work encompasses how the physicochemical factors determine phytoplankton biogeography and how, in turn, these organisms influence on the distribution of carbon in the ocean. She combines model, laboratory, remote sensing, and fieldwork observations in her work, which I have to say there is a very extensive list of papers and other contributions. And today she's going to talk about modeling phytoplankton responses to a warming world. Please, thank you.
Well, thank you very much. Thank you for the introducing me. Thank you very much to the organizers. I've actually always wanted to come to a Challenger meeting, so this is a great and what a beautiful place to have it, so thank you. The title is a little bit different to what was in the program, because I suddenly realized it was the first talk of the event, and maybe I should make it a bit broader than going into some really deep into ecological theories. So, modeling phytoplankton responses in a warming world. Am I supposed to point it anywhere in particular? Or? All right. So phytoplankton, microscopic organisms that photosynthesize by bringing sunlight into energy into the ocean, into their cells. They are the base of the marine food web. They're also incredibly important in the carbon cycle of the Earth. They fix as much carbon, produce as much oxygen as terrestrial plants. And they take some of the carbon, so they take carbon up in the surface ocean because they need sunlight, so take it up in the surface ocean. But some of the carbon in sort of dying cells, a small amount, makes it down to the deep ocean and is therefore forms a sort of reservoir of trapped carbon down in the deep ocean, which is also, again, important for our carbon cycle. The phytoplankton also, it's either too fast or too slow. Anyway, phytoplankton are also incredibly diverse. They have many different functions. Some fix nitrogen, and fixed nitrogen is actually limiting in many parts of the ocean. Some form silica shells. They also span several orders of magnitude in size. And so you can imagine these different functions and these different sizes support very different food webs and very different roles in that carbon cycle. So some export more carbon than others. So I think it's appropriate to want to understand what can happen as we warm our world. What will happen to these plankton as we warm our world? And so my talk is going to have several components to it. One is to ask, do we see any changes in phytoplankton at the moment? Do we know if phytoplankton are changing? Then let's dig a little bit more into what might actually be happening to phytoplankton. How would they change in a warming world? And then I will end with some perspectives of how we might actually do better in trying to be able to predict what will happen in a future world. So let's start off with the first concept is, can we see changes in phytoplankton in the world? Now, I think there have been many coastal or long-term time series which suggest that there may have been something that is consistent with phytoplankton changing due to climate change. But in this talk, I'm going to specifically be looking on a global scale. And therefore, the sort of appropriate thing to be looking at is satellite estimates of what's happening to the phytoplankton. So here I'm showing a movie from one of the NASA satellites of chlorophyll. So chlorophyll is the pigment inside phytoplankton that they need in order to be able to harness the sunlight. Check the color bar, though. It varies in different regions, across several orders of magnitude in concentration. And you can see that it changes a great deal over the course of the year, different seasons. What is not quite so obvious, as I'm showing this movie, is the incredible variability that is interannual or multi-year changes. And so it's quite difficult to actually detect changes on a long term of these plankton. So here, one of the satellites has been up. So the NASA MODIS Aqua satellite has been up just over 20 years now. And it seems appropriate then to ask, have we seen a change in phytoplankton over these 20 years? So this is showing the trend in phytoplankton, the trend in chlorophyll over those 20 years. Red meaning there's been an increase, suggesting a trend is increasing. Blue being the trend is decreasing. But the intensity of the color is actually the signal to noise ratio. So the signal here being the trend, so is it going up or down? It's a linear trend over those 20 years. The noise being the variability that I was mentioning, so interannual, multi-year changes. And statistically, if that signal-to-noise ratio is less than 2, it suggests that it's not significant. It could just be that the trend is no bigger than is not big enough to be definitely outside of that interannual variability. So we can mask out anywhere that had a signal-to-noise less than 2, and you can see very little part of the ocean is showing a significant trend. It's not to say that there isn't changes happening, it just means we can't detect it yet because the noise is so large. And this sort of idea is not surprising. Stephanie Henson did some work several years ago looking at model output. It suggested it might take several decades, 30 to 40 years, before we'd really be able to detect changes. But the satellites don't actually measure chlorophyll. What they measure is the amount of light that is being reflected out of the ocean. So that's the little LU. And one of the basic units of satellite measurement is the remotely sensed reflectance, RRS, and I'll use that quite often. And that is the ratio of what's coming out of the light coming out of the ocean relative to what's going in. And in particular, those satellites measured in different wave bands. So this is wave bands, think color. So blue, one sort of a low glow wave band would be blue, a high wave band would be red. And particularly, we're going to be looking in the visible spectrum. Why is remotely sensed reflectance important or useful? Because the stuff in the ocean changes how the light, how much light is being absorbed, and that LU is a sum of everything that's happened to that water, to that light, since it's gone into the water. It's been absorbed by things, for instance, chlorophyll. It's been scattered by particles, by phytoplankton themselves. And so what's coming out has got a lot of information about what happened in that ocean. For instance, in the bottom left there, I'm showing a spectrum of across those wave bands. So the x-axis is the wave bands from 400 to 700. So 400 being about blue, red being 700 being red. The x-axis, the y-axis is the remotely sensed reflectance, and I'm showing 3 different chlorophyll concentrations, water with 3 different chlorophyll concentrations there. As you can see, that there isn't much change in the green between these 3 different waters, but in the blue there's a great deal of difference, and unfortunately it's not showing up awfully clearly. But as you go towards higher concentrations of chlorophyll, there's less and less blue reflectance. And so you can use this information to then say how much chlorophyll there might be in the water. So how about we look at the trends in the remotely sensed reflectance rather than chlorophyll, which is which is already using those remotely sensed reflectance information. So here is the trend, and again it's been everything has been, the only colors you're seeing is whether the signal to noise ratio is above 2. And you can see, again, there's some places that are showing small amounts of change, but not a lot. So showing results here from a paper by B.B. Kale, where he decided, let's not just look at an individual signal, such as one remotely sensed reflectance, how about looking at them all together in a multivariate trend detection? So to kind of give you an idea here, I'm looking at, say, maybe 2 wave bands, beta 1 and beta 2. There's an uncertainty in the trend in each of them. But if we make a vector out of them and then look at the trend, there is actually less uncertainty. So now let's take all 10 of the modus reflectances and look at the multivariant trend. And we get a picture that looks like this. So now, again, the color is where there is a statistically significant trend. And it's showing up in about over 40% of the ocean. So that means 40% of the ocean has changed color over the course of the last 20 years, which is quite stark, kind of interesting. So the color of the ocean has changed. And then we get to a little bit of a chicken and an egg situation. Why has it changed? Well, it could have changed because something's changing in the phytoplankton. It could have changed for some other reason, but because phytoplankton are so sensitive to light, that if it changed for some other reason, phytoplankton would have changed as well. So whatever is causing those changes, we know that phytoplankton will have changed over the course of these 20 years. But the study doesn't say why it's changed. Okay, immediately came to mind that it's probably anthropogenically induced climate change, but we don't know that. We just know that it has changed after 20 years. And we don't know how it's changed because this is a multivariant trend detection, so it doesn't necessarily give us much information about how it's changed. And so we're going to come back to that later on in this talk. But let's move on to thinking about how phytoplankton might respond to a warming world. Okay, so we know the air temperatures are changing. We know that a lot of that heat is going into the ocean and warming it up. That's changing the amount of sea ice. The heat that goes into the ocean hasn't redistributed over the entire ocean. A lot of it's still in the surface ocean. And so that leads to increased stratification. That's going to alter some of the circulation patterns. And some of the carbon that we've released has gone into the ocean, about 30% of it. That's changing the pH of the ocean, so ocean acidification. How are those likely to affect phytoplankton? Well, first of all, the warming temperatures are likely to lead to faster biological rates. So here is some synthesis of a bunch of laboratory experiments. Each of those lines is a different diatom, which is a type of phytoplankton, and that have been grown over different temperatures. And so each line is how that diatom responds to different temperatures. You can see there's an optimum temperature. But what I want to draw your attention to is the blue line, which is a fit to the envelope of all of them. And it shows that warmer adapted diatoms tend to grow faster than colder adapted diatoms. Similar sort of increases biological rates happen with other things like zooplankton, bacteria. And so in general, warmer temperatures are going to lead to faster rates, biological rates. Changes in sea ice and the increase in stratification are going to change the light environment. And again, the light environment is really key for the phytoplankton. Changes in stratification, change in circulation are going to change the amount of nutrients that make it to the surface ocean. Some of those nutrients that phytoplankton need, like nitrate, are more abundant in the deep. And so as we change circulation and we change the stratification, we're changing how much of that nitrate can make it to the surface, we're going to have likely a decrease in the amount of those nutrients available in a future warmer world. So how might all of those together, so the many stresses happening, how are those likely to affect the phytoplankton? We're particularly going to answer that question by looking at a mechanistic model. And by mechanistic model, I mean one where we're using the best knowledge of of the system to try and suggest how something might change over time. And I just want to sort of contrast that, for instance, to a statistical model. So here we're not necessarily using data per se. What we're using is the best knowledge that we've gained from the data in order to construct the models. So we might look at a system like this where we're looking at matter as it goes from nutrients, is taken up by phytoplankton, is eaten by zooplankton. They're going to produce detritus, and the detritus is going to be remineralized back to nutrients. So we can ask, how does each of those pools, how does each of those boxes change over time? So the change in phytoplankton would be something to do with how it's growing, and that's going to be in response to the nutrients, the light, the temperature. The losses is going to be things like grazing, viruses, other cell death. And then all of this is in a moving ocean, so how are they changing because they're being moved around? And what we can ask is, what we can then say is that if we know the biomass at time 0, then sometime later, it's just the change in biomass times the amount of time that has happened between them. And so we can construct a set of equations that look through at these changes over time, and we can turn that into computer code and let a computer do the stepping forward in time for us. So from one time to another, to another, to another, over many, many years. And we get from that three-dimensional depictions of how things are changing in time. So 3 dimensions, lat, long, depth. And so we put this all into a model. The model then includes the physics, so how things are being moved around, the biogeochemistry, so that's the cycling of things like carbon, nitrogen, phosphorus, iron, and the plankton themselves, so the phytoplankton, the zooplankton. Here we're showing chlorophyll concentrations as produced by this model, producing a world that seems reasonable. We're seeing the changes over seasons, the patterns of the sort of subtropical gyres with very low chlorophyll, and these higher latitudes with higher concentrations of chlorophyll. And we can use a model like this to then address this question: how will phytoplankton respond in a changing ocean? So we'll take that ocean and we will subject it to a warming world. So we're going to look at the worst case scenario. Well, let's hope that it's the worst case scenario. So it's the pink line there, a very, very high emission scenario where we have made no effort to try and constrain our greenhouse emissions. And you can see on the bottom graph there what it would suggest about the changes in air temperature over the course of the 20th century. So almost everything I'm going to be showing from now on is going to be the results at the end of the 21st century in this really high emissions scenario. Keep that in mind because, as I said, this is hopefully the worst case scenario. I also wanted to point out that I'm not actually going to run a model that's of that high resolution for 200 and something years, because that would be computationally expensive. So I'm showing this because it has all those beautiful eddies and excitement. But in fact, we're going to run a much coarser resolution model. And I'm also going to show you a bunch of different studies with slightly different versions of the model. Just another thing to keep in mind if you're like, oh, that one didn't quite match that one. They are slight variations in the model. Okay, so what does the model suggest will happen to the phytoplankton in a warmer world? So we know that there are multiple stresses. How do those interact with one another? So this is results from a simulation where I'm looking at the total, the globally integrated primary production, so that's the amount of carbon the phytoplankton have fixed, and I'm looking at the difference between actually a control run, so that's where there's been no emissions at all, so that it's a sort of stable run, and one with this high emission scenario. The black line is how the globally integrated primary production has changed over the course of the 21st century in this simulation. But because it's a model, I can pull apart how much is due to warming, so that's the red line. Because of these increased biological rates, that would lead to an increase in primary production. But because there's also a reduction in the amount of nutrients making it to the surface, that would lead to a reduction in primary production, and the total is something in between. But it's not. But what's important is that these are going to be happening in the decrease in nutrients versus the temperature changes are going to have different impacts in different places. So you get a geographically very different, very complex pattern. You can see in some regions which had low nutrients, it's likely to be a reduction in primary production. But in some places, in higher latitudes where there were a lot of nutrients coming to the surface, these higher biological rates are more important, and you're getting an increase in primary production. So I'm showing you results from one of my models, but I'm certainly not the only person who is trying to model what's happening to phytoplankton in the future ocean. So here are some results from the CMIP-6, and so that's the Coupled Model Intercomparison Project that is used to inform the IPCC. There are 13 different models that included ocean biogeochemistry and ecosystems. So on the left is the mean primary production from all of those models. On the right is the changes that they predict in this high emission scenario by the year 2100. So purple meaning a decrease in primary production, green being increase. Some similarities to what I saw in my model. But what I want to draw your attention to is the stippling. So that's the little dots that you're seeing, like along the Southern Ocean, a little bit in the North Atlantic. Those are where the models agree, 80% of the models agree on the sign of the change. So I'll just let that settle in for a bit. Anywhere that there isn't a stipple, it means that the models have different ideas of whether it's going to be positive or negative. So we're rating, what's this, the phase 6 of these coupled models, and the models are still not actually sure where we're going to have increases, where we're going to have decreases. This is not to knock the models. This is an incredibly difficult problem. But we can maybe make some suggestions that some low latitudes are going to have decreases, some high latitudes are going to have some increases. But it's certainly something that we need to understand better in order to be able to predict what is going to happen to productivity in the future. So I'm more interested actually, though, in how the phytoplankton diversity is likely to change in this changing world. So how are these different stresses likely to alter the communities of these diverse phytoplankton in the future world? And so our model is actually developed precisely to look at that sort of question. So here I'm showing a schematic of our model. It doesn't just look at one group of phytoplankton, but a whole bunch of them. In the row, there are different size classes. In terms of ESD, which is the equivalent spherical diameter, so it's as if we took any phytoplankton and made it round and then looked at what its diameter was. So going from 0.4 microns up to about 100 microns in the phytoplankton. In the different columns are different functional groups, so whether they fix nitrogen, whether they form silica shells. The mixotrophic dinoflagellates are both grazers and can photosynthesize. And we have a set of heterotrophic zooplankton that eat the phytoplankton. So the arrows in there are who eats whom. The nice thing about developing a model along these sort of traits in particular size is that we don't try and decide what the parameters are for what the maximum growth rate, etc., are for each individual of those little bubbles, so each individual phytoplankton group, but rather we use empirically understood parameters that are based on size. So we know that something that is a certain size will grow faster than something a little bit bigger than it. And so we use these allometric scalings in order to be able to parameterize this with not as many parameters as you might expect for that number of phytoplankton. And then we can look, for instance, at how the model, we can evaluate it against how we might expect the community structure to be patterned. So on the right here is an estimate from satellite of how different size classes, so pico, nano, micro groups should be distributed. And on the left is the model. And we're capturing the fact that the smaller phytoplankton, the pico, are more ubiquitous, and then the structuring becomes more defined as you go towards the higher to the higher nutrient regions with the larger size classes. And so we can ask, how does this size class change as we go into the future in these high emission scenario? So here is the percentage change that the model predicts by the year 2100. And see, some regions become slightly more, that's where it's red, but a lot of the ocean shows lower in all size classes. But in particular, notice that the smaller phytoplankton change less than the larger phytoplankton. So there's more blue and deeper blue for the larger size classes. So this is some work that Stephanie Henson spearheaded. So she looked at the size spectrum of these changes. So suggesting that in most cases there are more small phytoplankton than large phytoplankton. So that's what I've schematically shown, biomass against size. And where in that map on the right is blue, it suggests that the spectrum has decreased, and so that you're getting more small phytoplankton than large size phytoplankton. And the majority of the globe is showing that decrease. So what does it mean? Larger phytoplankton are in general declining more than small phytoplankton. And I forgot to mention the main reason for that is the smaller phytoplankton are better adapted to lower nutrient conditions. So as we go into lower nutrient conditions, they're going to be responding slightly less badly than the large plankton. But also kind of interestingly, in that, for instance, in the Southern Ocean where we predict an increase in biomass, there's actually going to be more small phytoplankton than they're actually going to grow in more than the larger phytoplankton. And that's going to have, these sort of changes are going to have a consequence to the higher trophic levels, to the zooplankton that would be eating those phytoplankton. The smaller phytoplankton are going to be supporting a very different food web to the larger phytoplankton. We can also look then at the functional groups, so that's looking on these columns of this model, how different, for instance, diatoms, so the silicifiers, might change in the future. So here I'm just showing the sort of current day model predictions, and then against some data synthesis to show that we're getting some of the patterns of, again, a more ubiquitous picophytoplankton and the more structured as we go to different functional groups. So changes in the future. Again, this is showing the percentage change in the biomass in the phytoplankton. Again, we see a lot of the low latitudes having a reduction in almost all types, but we see some increases in the higher latitudes. In particular, we see some changes in the range of some phytoplankton. So for instance, the diazotrophs, so that's the middle column. And diazotrophs are kind of important because they fix nitrogen, and fixed nitrogen is really in limited supply, so they're actually important in being able to support other phytoplankton by producing this fixed nitrogen. The model suggests that there will be an increase in their range, and this is due to changes in the amount of nitrogen and of nitrate versus iron that is available in the future world. Now, you had to look at lots and lots of different little maps, but let's try and put it together in one metric. So again, this is works spearheaded by Stephanie Henson. She looked at the turnover of the community between the beginning and the end of the century. So the turnover is the number of types gained or lost in any location relative to the total number of plankton that are there. And so you can see in many regions there are 15-20% change in the type of phytoplankton that are in any location. So a large change in the community structure. So a take-home from there, different groups respond differently. There's significant changes in community composition. And again, this is likely to have a very large impact on higher trophic levels. So I've said that twice now, so actually, since I do resolve zooplankton in the model, let's go and have a look at those changes. So here I'm showing on the left the annual biomass of phytoplankton on the top, and their grazers, the next trophic level, on the bottom. And on the right is the change by the year 2100. the point to bring here is that in some of those lower latitude regions, the autotrophs have decreased, but the grazers have decreased even more. So because their prey has decreased, and they're on a case where there is actually very little prey, any small amount of their prey leads to a larger change in those grazers. And this appears to go up through the food web. There's some, I'm listing there a bunch of other papers that have looked even further up that have looked at this level, or even up to fishery, and suggesting this trophic amplification, the fact that in some places there's a larger change in the next trophic level up, has been sort of suggested. So I've spent a lot of time talking about the sort of in-depth details of a complex marine food web, complicated phytoplankton. But let's bring it back to the question we had earlier about detecting changes. So let's go back to this study, which is, again, showing actual changes from actual satellites of the real ocean and how it's changed over the last 20 years. And ask, what caused this change? Is it anthropogenic? Well, the nice thing about the model is that it also includes a radiative transfer. So that is, we also model how light comes into the ocean and is absorbed and scattered by the stuff in the ocean. So that's the phytoplankton, the detritus. We resolve it on several different wave bands. So that's what we have at the bottom. And we can ask, how is the remotely sensed reflectance changing over the course of the 21st century? So this is showing the change in at 475 nanometers, so that's sort of this green-blue color. Where there is color, it means there's a significant change. And when we can see a lot of the ocean has a significant change in the remotely sensed reflectance at this wave band. So Kale took the remotely sensed reflectance over the full wave bands, did the multi-variant trend analysis on the model data at each year in the model as we go from beginning of the century till the end of the century. And that's the green line in this graph on the right. And what we're showing on the x-axis is how much of the ocean, the fraction of the ocean that is showing a significant change over the course of that 20th century. So by the year 2100, the model is suggesting about 90% of the ocean is going to be showing a significant change in color due to anthropogenically caused climate change. And we know that in the model because we've run this baseline where we haven't changed emissions and there is no change in the color. And then this case where we have run the model with increased emissions and increased warming and all the other stresses that are happening, and we're seeing this change. The model suggests by about 20 years that about 40% of the ocean will be showing a color change, which actually quite remarkably matches up with what Kale found from the real world. And so it suggests that these trends that were seen in the MODIS satellite are consistent with climate change by climate change induced changes in the ocean. And that gives me some pause. I mean, we all do science and we all talk about climate change, but every now and again you go, oh my goodness, we have caused 40% of the ocean to change color already. And if we don't change what we're doing, the whole ocean is going to change. The changing of the ocean, color of the ocean affects the phytoplankton or is affected by the phytoplankton. Either way, it means we have changed what is happening to the phytoplankton over the open ocean, way out in the middle of nowhere where only satellites are really able to predict this. Because we have the model, we can also begin to look a little bit more into what those sort of changes are. So this is some work that Francesca Mattai, who's a postdoc in Villefranche, is looking at. He's run the same sort of model, particularly interested in how the color is changing. And so what we're showing here is the blue to green ratio within the water column. And so where the ratio is higher, there's more blue light. So where it's bluer is bluer. And Where it's greener, there is less blue relative to green. And now, if we look into the end of the 21st century, which is what he's showing at the bottom, that's the change in this ratio. And very confusingly, it's plotted so that where it's become bluer, it's redder. So where it's redder, the oceans become bluer. Where it's blue, it's become greener. And what we find is that we're actually getting to be a much stronger gradient between the blue and the green regions. So the transition zones between the gyres are growing smaller. Some of the regions that are low in productivity are tending to go lower, and some of the ones that are higher in productivity are going greener. We can also look at, for instance, what's causing some of those changes. And I'm not actually going to be able to give you the exact answers to why, exactly what is changing, but we can begin to pull it apart a little bit. So here's changes in chlorophyll, % per year. But then let's also look at how other pigments are changing. So phytoplankton don't just have one pigment, not just chlorophyll A. They also have a bunch of other different pigments, some of which are also helping in photosynthesizing. Some of them, like the non-photosynthetic ones in the bottom right, are ones that are actually more like sunscreen. They're trying to protect them from too much light. And again, what I'm showing here is the percentage change over the course of the century for each of these different pigments, these accessory pigments, and you can see they're actually stronger than what the chlorophyll changes are. Some of it is because some phytoplankton, for instance, the ones with chlorophyll B, not all phytoplankton have chlorophyll B, and so what you're seeing in the red there is where they're expanding their range. But it suggests that the signal of community structure change is going to be stronger than the changes in a bulk product like chlorophyll A. These pigments have very different absorption capabilities, so that's shown on the right. Chlorophyll A is the dark blue, but the different colors, you can see they're absorbing in very different colors. And so that is going to what's actually causing a lot of the change in the color of the ocean. I don't have time to go into each of the individual, but there are other things in the water like colored dissolved organic matter, which absorbs really high in the blue. That seems to be changing a great deal too. Particles scatter very, very differently depending what size they are, so that too is going to impact the color change. And so that's stuff it's difficult to pick apart, and things that we're slowly doing in the model, and that's some work we're doing with Anna Hickman at Southampton. Okay, so I haven't actually quite finished the talks, but this is a summary as so far, just so we're up to date. There is a change in the color of the last over the last 20 years, and it seems to be consistent with changes that would occur because of climate change. And that is likely going to be due both to changes in biomass of phytoplankton, but also to changes in the community structure. We've shown that there are many stresses happening to the phytoplankton. Their whole world is changing, the light is changing, the temperatures are changing, the nutrients are changing, and some of these are acting in opposite ways, leading to a very complex pattern on how productivity will change, but also how community structure changes. Small changes in the competitiveness of each of different phytoplankton is going to lead to large changes in community structure. And all of this will lead to complications further up the food web. So even if you didn't really care what's happening to the color of the ocean, the phytoplankton, you might care what might land up on your plate when you go out to eat tonight. And that does really matter what is happening to the phytoplankton. So I'm going to end off with some sort of perspectives on what might be helpful in looking at climate change-induced changes. Sorry, changes twice. But anyway, how we can better predict what will happen to phytoplankton in the future. So some of those are to do with monitoring. As we showed, 20 years of a satellite is beginning to show changes in ocean color, but we need longer monitoring to see changes in things like chlorophyll. And so that's the importance of continued satellite measurements, but also intercomparability between those satellite sensors. The satellite sensors that most of the ones we have up in space at the moment have very few wave bands, so they're only capturing light at a few colors. And we really need a much wider breadth of wave bands in order to be able to detect community structure changes. So NASA just recently, sort of several months ago, launched PACE, which is a hyperspectral satellite mission, and this will hopefully be able to allow us to really be able to detect community structure changes going forward. We've spent the entire of this talk talking about surface, what's happening at the surface, but the model also suggests that actually some of the bigger changes are happening deeper down, especially as mixed layers shoal and sort of deep chlorophyll maximums might shoal. And so it's important for us to be able to monitor deeper down in the ocean, and why things such as bioargo or LIDAR, which could be on satellites and look deeper into the water column, will be really important going into the future. We stressed how the phytoplankton we showed how the phytoplankton are dealing with a lot of stresses, and it's important to know how those stresses are interacting with each other on the organism at the physiological level. And so the importance of continued laboratory studies looking at, and mesocosm studies that look at such things. From my perspective, we've shown how difficult it is for models to be able to make predictions. And so there are a number of ways that we can maybe, as we're going forward, try and help models. And so I'll spend just a couple of minutes saying that, yeah, we've showed that the models are having a difficult time, but we shouldn't throw up our hands in the air. We also shouldn't probably move to looking at looking glasses, and we should be actually trying to make an improvement on how we study these things. So we already showed that the CMIP models are having a great deal of difficulty agreeing with one another. Now that's due to many reasons, like the differences in the physics in those models, the difference in how the biogeochemistry is done, but also how each of those models parameterizes how things like nutrients and temperature affect the plankton. And just to give you a sort of small feeling of that, this is some work by Stephanie Anderson that she did while she was a postdoc at MIT. And this is how do we model how phytoplankton respond to temperature. The dots on all three of these plots are the same. They're from laboratory experiments where phytoplankton have been grown at different temperatures. And a typical way of looking at these things is to decide what a maximum growth rate is, and that's what we'd put in a model. And that would be the black line that is shown on the EPLI. So if you assumed all phytoplankton were the same, and we wanted to model them as just one single entity, we'd use that black line. But if we wanted to accept the fact that there are different types of phytoplankton, so we said there's like diatoms, there's cyanobacteria, do they respond differently? We could do something like the middle one, where we assume they all respond the same to temperature, but with different maximum growth rates. And so now you can use the dots in there and fit lines to them. Or, as Stephanie Anderson suggested, that in fact maybe different types of phytoplankton respond differently to temperature. And so you can see these crossings of these lines. And if you run a model with three different models with different ways of looking at the temperature, you get very different results and different suggestions of who will win in a future world and who won't. Especially when you look at, for instance, on the right one, the coccolithophores, the yellow is as the laboratory results suggest there's a very low slope. And so, whereas something like cyanobacteria seems to have a very, very high slope. And so the competition between those plankton will be quite different in a future world. Another thing to point out in the CMIP models is that and these IPCC level models is none of them take into account the fact that the ocean is changing in its pH, that ocean acidification is happening. And the reason for that is that it's really not easy to be able to say what's happening because of ocean acidification. I tried to find one figure to kind of encompass what we know about it. And the best I could do was a schematic. So this is just a schematic showing how growth rates will change relative to PCO2, showing that most phytoplankton will have an optimal PCO2, but when the PCO2 is too low, they're actually starved for carbon. But that as you go to too high carbon, there might be problems with ocean acidification. And what the schematic is trying to show is that there is a very large taxon-specific sensitivity. In general, we think maybe that a small increase in PCO2 might lead to at least some of the phytoplankton having a higher growth rate. And if we put that into the model, that would suggest an increase in productivity. And so I've showed something similar before. That might just tilt this total productivity, the black line, from being negative to positive in the future world. But we really don't know enough. There's so much uncertainty in those parameters. But until we have a better certainty, we can't put it into models. And until we put it in the models, we can't really get a feel for what will really happen in a future world. There's a lot of difficulty also in how we manage to capture what's happening because of different changes, like what will happen if we have, for instance, on this, here we have maybe how a phytoplankton might change with temperature. Its growth rate will change with temperature. On here, this is growth rate here and how it might change with nutrients. Most models just take those two and multiply them together, but that's actually not what's likely to happen. There's probably likely to be very different responses to temperature changes at low nutrients than at high nutrients. And so having experiments that look at the multifactorial responses are really important. And I just want to give a plug for the SCORE working group, which is trying to set up and encourage experiments that just do look at just at that multifactorial changes. I think we started a little late, right? Okay, all right, thanks. Another thing that we have a great deal of difficulty capturing in models is actually how the grazing occurs, because it's really difficult to get the parameterizations and the sort of values for what maximum grazing rates should be, especially when looking at an aggregate of zooplankton. This is a recent paper which shows the CMIP6 models again. So if we just look at the ring on the right, this is the different CMIP6 models. And the author has depicted the different complexities of those different plankton. So we go from models that have just a single phytoplankton and a single zooplankton to maybe several phytoplankton to maybe lots of phytoplankton and several zooplankton. And I really like this paper, but I couldn't find one image from it that really gave you a good feeling for the results. So just sort of don't look too long, but listen to me instead. We're looking at grazing pressure on this axis and latitude, and each of these lines is a different model. And so the thing to take home is they're all over the place. They actually did include one of my models, and it's just nicely sitting here right in the middle, but not necessarily the best. So these grazing rates, how the grazing is done is really important. But there are other interactions that are also really important in setting where phytoplankton, their ranges, and things like interactions with bacteria, interactions with viruses, and I'll give a plug for Chris Follett's talk in a little while, which is looking just at how those might impact the ranges of different phytoplankton. And then on top of all of this, phytoplankton have generation times of a few days. And so the potential for evolution is very large. Are they going to be able to evolve into this new world? And there's a nice paper by Ben Ward, which talks about whether we should include, when we should and when we might not need to include evolution in our models. Just to give you a sort of little taste of that, here is an example of, say, two phytoplankton X and phytoplankton Y. And this is temperature and their growth rate. Y is a warmer adapted one. So if we look here, this is the biomass of those two phytoplankton in the year 2000 from one of our models. And you can see there is actually, the world is too cold for Y at the moment. But as we go to the future world, you can see that the phytoplankton Y is now taking over some of the range of phytoplankton X, and X is moving more poleward to where the water is more adapted to it, as that water is warming. But the question is, would species X be able to evolve? So which is happening quicker, Y evading its space or X evolving? And that's not necessarily obvious, and obvious, something that we need to think about as in the future of modeling. So I want to just end with acknowledging a great number of people who have been part of these studies. The movie has nothing to do with climate change. It's just a pretty movie, and it's just showing how complex the community structure is. Each color is a different combination of phytoplankton that exists within the model, and how the mesoscale, which I haven't dealt with at all, is really affecting a lot of this. And so with that, I'll take any questions with whatever time remains.